Assalamualaikum warahmatullahi wabarakatuh. My name is Sajidah binti Tajidin and my group members are Syahadah Abdul Malik, Shafiqah Ghazali and Amiera Izzah. So today we will be presenting about the chemiluminescence immunoassay. First of all, we will start with the principle of the chemiluminescence immunoassay. CL or chemiluminescence is defined as the emission of electromagnetic radiation caused by a chemical reaction to produce light, while the immunoassays is the test based on the specific binding that occurs between an antibody and antigen that is specifically recognizes. Chemiluminescence immunoassay is an assay that combines chemiluminescence technique with immunochemical reactions. Chemiluminescence immunoassay is a method to determine the concentration of samples according to the intensity of the luminescence that a chemical reaction emits. Chemiluminescence immunoassays also used to estimate those analytes which have extremely low concentrations in the blood, such as hormones, serological markers, and drugs. The principle of the chemiluminescence immunoassay is in the presence of the complementary antigen and antibody, the paratope of the antibody binds to the epitope of the antigen to form an AG/AP or an immune complex. Then, estimating the levels of such immune complex by use of labeled antibodies form the basis of CLIA. It also involves use of the stationary solid particles coded either with the AG or AB of interest. Post-incubation, which ensures intact immune complexes are formed, substrate is added. This results in generation of light, the intensity of which is directly proportional to the amount of labeled complexes present and which indirectly aids in quantification of the analyte of interest. The intensity of light is measured in terms of relative light units, RLU.
Okay, let's talk about labels. There are 4 points that I get for labeled chemiluminescence immunoassay. Reagents required for reactions that produce CL may be coupled to ABS or antigen and used as a label for IA. Since the first report on CL labels in 1917, this category involves labels that are consumed in CL analytical reaction, for example, luminol derivative, acronidium, esters, and NPS. Luminol is the best known and one of the most efficient CL reagents. It is coupled to ligand via reactions involving the amino acids. However, the resulting conjugates have lower CL efficiency than the parent compounds. Labels derived from isoluminol have been more successful. What are the labels used in immunoassay? Labels commonly used in immunoassays include radioisotopes, fluorophores, and enzymes. Signal detection methods include radioactivity, colorimetry, fluorometry, chemiluminometry, and nephelometry. Special immunoassays not requiring specific labels include immunophilometry, immunoturbidimetry, and agglutination.
Next, let's take a look on its classification. There are 3 types of chemiluminescent immunoassay technique. Sandwich assays. Microparticle reagent is mixed with the patient or control sample. These microparticles are parametric and coated with capture molecules, which is immobile antibody. Reaction mixture incubates an analyte present in the sample binds to corresponding capture molecules on the microparticle, forming the immune complex. A magnet attracts the parametric particle, now bound to the specific analyte, to the wall of the reaction vessel. Washing at this stage removes all unbound materials. A chemiluminescent acronidium label conjugate is now added, which bind to the immune complex to complete the reaction mixture, After a period of incubation, reaction mixture is again washed to remove unbound material. Pre-trigger is added next. Pre-trigger creates an acidic environment to prevent early release of energy or light emission. Trigger solution is now added to complete the reaction. The resulting chemiluminescence reaction is measured as relative light unit. A direct relationship exists between the amount of antigen or antibody in the example. Relative light unit was detected by architect system optic. The generated signal is directly proportional to concentration of analyte and computed accordingly. A competitive binding assay typically measures the binding of a labeled ligand to a target protein in the presence of a second competing but unlabeled ligand. This assay can be used to access qualitative binding information as well as relative affinities to or more molecules for one target. Direct assays at via XPT Center is a system that directly measures the light's energy without use of added steps or amplifying molecules. Assays use acronymium ester as the chemiluminescence label because it's not require the addition of catalyst or substrate. XPT Center use the dimethyl form of acronymium ester because its stability allows long region shelf life.
Example of test. A standard example of chemiluminescence in the laboratory setting is the luminol test, which luminol chemiluminescence test for blood. The luminol chemiluminescence reaction is responsible for the glow of light stick. The reaction is used by criminalists to detect traces of blood at crime scenes. How it works? Blood is indicated by the luminescence upon contact with iron in hemoglobin. When the chemiluminescence takes place in living organisms, the phenomenon is called bioluminescence, which a light stick emits light by chemiluminescence. To make the luminol solution, luminol powder C8H7O3N3 is mixed with the hydrogen peroxide H2O2 and a hydroxide, for example KOH, in a spray bottle. The luminol solution is sprayed where blood might be found. The iron from the hemoglobin in the blood serves as a catalyst for the chemiluminescence reaction that causes luminol to glow. So, a blue glow is produced when the solution is sprayed where there is blood. Only a tiny amount of iron is required to catalyze the reaction. The blue glow lasts for about 30 seconds before it fades, which is enough time to take photographs of the areas, and the area can be investigated more thoroughly. Thank you for listening to our slide presentation.